Okay, let's unpack this. If you think of a cell as like a sophisticated miniature city, every internal structure, every organelle, it has its own unique molecular walls, right? Fences. For decades, the main idea, the dogma really, was that materials to build and maintain those walls were delivered slowly, using bulk transport. Think of them as these massive, slow-moving shipping containers.
Vesicles.
Exactly. That was the standard picture. But this new research fundamentally challenges that. The single most important finding here, I think, is that the cell city relies almost entirely on, well, high-speed express lanes, you could say. Organelle identity seems to be maintained not by that slow, sort of non-specific vesicular transport.
Right, the bubbles.
Yeah, the bubbles. But by incredibly fast, selective, non-vesicular transfer. And this new route, it's up to 11 times faster.
11 times. That kind of speed difference is massive. And, it starts to explain a puzzle cell biologists have had for years. Lipid transport is absolutely foundational to everything. But cells have, what, over 1,000 different kinds of lipids?
That's right, a huge variety. So how do the right lipids get from where they're made, often the endoplasmic reticulum, the ER, to where they need to go, like the outer plasma membrane or PM? And crucially, how do they get back? Was it always those slow conventional bubbles, or was something else going on, maybe being handed off by specialized proteins, the lipid transfer proteins or LTPs at these contact points? We needed a stopwatch.
We needed a stopwatch, yeah, but one that was also a kind of chemistry lab. The real innovation here wasn't just seeing the transfer happening, it was finally putting quantitative kinetics behind it. Numbers, rates. And to do that, researchers developed what they called bifunctional lipids. These molecular probes are, frankly, the game changer.
Okay, tell us about these probes. Why bifunctional? What makes them so revolutionary here?
Well, they basically solved a critical measurement problem. These lipids have two key functions. First, they're photoactivatable. That means researchers can pulse a light and instantly turn on the label for the lipids only at a specific place, like the plasma membrane, and at a precise moment. Control over space and time.
Okay, so you know exactly where and when they start.
Precisely. And second, they're clickable. That means later, after the experiment's run its course, they can use click chemistry and then ultra-high-resolution mass spectrometry to see exactly which specific lipid species moved, where they ended up, and even what they might have metabolized into. It's like a spatial and temporal GPS system for these molecules.
A molecular GPS. I like that. So once they put that GPS system in place and actually tracked the movement, the speeds were just staggering.
Absolutely staggering.
Yeah.
By tracking how fast these labeled lipids moved from the plasma membrane back towards the ER, that's the retrograde direction, they found that this non-vesicular transfer route was up to 11-fold faster than the vesicular route going towards endosomes.
11-fold. So it's not just a slight preference.
Not at all. This kinetic data strongly the numbers suggest that non-vesicular transport is responsible for something like 85 to 95% of the PM lipid transport in that retrograde direction.
Yeah.
It's the dominant high-speed highway, period.
Okay, but that raises a question for me. If non-vesicular transport is basically just these lipid transfer proteins handing off molecules at membrane contact sites, almost like a simple pass-the-baton action, what stops those molecules from just diffusing randomly back and forth across that connection point? What enforces the direction? How does it ensure flow towards the ER?
That's a really critical point.
Yeah.
That speed, it raises a fundamental control issue, right? Mm-hmm. You can't have chaos if you want to maintain these distinct organelle identities. And the answer, it seems, lies in energy coupling. They found the directional flow is actually driven by active ATP-dependent flipping.
Flipping? You mean like flipping a coin?
Sort of, but with lipids across a membrane. Think of a protein called a flipase, specifically, a type called P4-ATPases. These flipases use the energy from ATP to actively move specific lipids from one side of the membrane leaflet to the other. It maintains asymmetry.
Okay, so they use energy to move lipids side to side within the membrane. How does that direct traffic between organelles?
Great question. The researchers tested this directly. They used genetic knockdown to reduce the levels of a key flipase subunit called TMM30A. Now, when they slowed down that flipper activity at the plasma membrane, the transport rate of PE, which is a common lipid species they tracked, heading towards the ER, slowed down by threefold.
Threefold slower just by messing with the flipase.
Exactly. It's direct evidence. It suggests the flipase acts almost like an ATP-powered turnstile or maybe a bouncer at the PM exit. It grabs the lipid, uses energy to flip it, maybe presenting it to the LTP, and crucially prevents it from just slipping back out to the plasma membrane once it's been handed off. It enforces that directionality.
Wow, so it's a beautiful coupling of the handoff by the LTP and this powered turnstile,
Right.
flipase. And they took it even further, right? Connecting transport speed to the bigger picture of cell composition, they compared these incredibly fast transport rates to lipid metabolic rates, how fast lipids are actually made or broken down locally.
Yes, and what's really fascinating here is just the sheer difference in scale. The transport kinetics, the movement speed, was generally found to be 10 to 60 times faster than local metabolism.
10 to 60 times faster.
Yeah. So think about what that means. The composition of an organelle, what makes the ER membrane distinct from the PM membrane, it isn't primarily dictated by slow local construction or demolition, making or breaking down lipids right there. It's determined by how fast specific materials are actively shuttled between compartments. The cell's identity, in terms of lipids, seems defined by its express shipping network and its speed.
That really shifts the perspective. So this whole pipeline they developed, it provides the missing quantitative framework for understanding these contact sites, doesn't it? It's not just showing, oh, look, transfer happens. It's providing a rigorous reference map for the actual rates. That clarifies how cells truly maintain their specific environments.
Precisely. It's a quantitative map. And we anticipate this will have major implications for understanding various disorders that are rooted in lipid trafficking problems.
Yeah.
Things involving the lysosome, endosome, ER circuits.
Yeah.
Now, researchers have a solid kinetic baseline, a map, to start forming and testing hypotheses about therapeutic targets.
That is a true deep dive. Okay, let's quickly recap the 3 key takeaways listeners should have from this. First, retrograde lipid transport back from the PM is overwhelmingly non-vesicular. It's way faster, up to 11 times faster than the traditional vesicular traffic.
Right. That's number one.
Second, this fast directional movement isn't just passive, it's energy coupled. It's driven by these ATP-dependent flipases working alongside the lipid transfer proteins, acting like powered turnstiles.
Got it. Directionality needs energy.
And third, organelle membrane identity. Its specific lipid makeup is primarily controlled by these rapid transport kinetics, which are much, much faster, 10 to 60 times faster than local lipid metabolism. Shipping speed defines the neighborhood.
Absolutely. Those are the core findings. And maybe here's the thought to leave you with. If the cell is so reliant on these ultra-fast protein-mediated transfers at contact sites, the next really critical question might be, how much does simple physical separation between organelles actually matter? Could a cell potentially reorganize its internal geography, maybe move organelles closer or further apart to favor one type of transfer over another? Or is this architecture pretty much fixed? It suggests a whole new layer of control, maybe spatial control, that we now need to explore.
Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize-winning lab. This channel has 3 main goals. 1, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. 2, to build a supportive community where we can exchange the most valuable experience-based research advice. 3, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the 3rd goal. Let's go! Let's make great science together.